I'm Sri Narasimhan, scientific editor at Cell, and you are listening to a Cell paperclip. Today we're chatting with Dr. Elaine Lorenzen from the University of California, Berkeley, about polar bears, brown bears, and all sorts of interesting findings that come from a population genomics approach. Hi, Elaine. Thank you for joining us.
Yes, thank you.
Could you tell us a little bit about your background?
So I'm a biologist, and I've worked both in population genetics and ancient DNA on large mammalian megafauna species, both extinct and extant, over the past probably about 10 years. Worked both on African megafauna systems and extinct northern hemisphere megafauna, now in polar bear as well.
So what kind of approaches do you use for your studies?
So I use DNA and population genetics, and in the case of the polar bear work that we have here, it's population genomics, where we use DNA to infer demographic histories and selection and adaptation in the genome.
In your Cell paper, again, this is a population genomics approach to studying evolutionary adaptation in polar bears. So what was really the motivation behind this study?
You know, when we started out, we didn't know how old the polar bear was as a species. There'd been a couple of publications which had findings which spanned from 600,000 years ago to 5 million years ago. So we had no idea how old it was. And then it's a species which shows it has an incredibly specialized way of life. It shows extreme adaptations to life in the high Arctic. And to try and understand the genetic mechanisms that make that life possible, polar bears have such a high fatty diet. They eat a lot of seal, primarily marine mammal prey, a lot of seal blubber, and how can they cope with that? So it was an interesting evolutionary wildlife model system for evolutionary adaptations to extreme lifestyles. The closest living relative of polar bears are brown bears, so we used a comparative genomic approach where we looked at several brown bear individuals and the genomic diversity within that species, and then we compared it to a large population of Greenlandic polar bear. And then we looked at, first of all, what point in time did the species diverge? And we found we see that polar bear is only under 500,000 years old. And then we looked at the genes in polar bear relative to genes in brown bear, and then we looked specifically at which genes showed a very strong signal of selection. Then we looked through those genes, the top candidate genes, and then we found some very interesting candidates, including genes where we know that variants in the gene change coat color to a lighter coat color. And we also found genes associated with cardiovascular diseases in humans. And then we found a gene which is very interesting, which aids in the removal of lipid from blood and lymph.
It's really intriguing that this adaptation is, in a sense, remarkably recent, and also the adaptation, like a large fraction of it, is metabolic in nature.
Yeah, it was incredible, actually. I don't think in the studies, to my knowledge, that have been published so far where we look at adaptations and the functional categories which are enriched for novel environments. Normally we see immune-related genes. I don't think, to my knowledge, that there's been a study which shows these genes associated with cardiovascular diseases and with adipose tissue development, which really makes sense in a species where fat is so important. Not only do they have a large layer of blubber under their skin, but they also, of course, ingest fat even from babies. The milk of polar bear mothers is up to 27% fat. And then after that, they have primarily marine mammal prey, and a lot of it is seal blubber, what they eat. So it really made good sense of data. So we were very excited about that.
So I'm curious, how does sampling work? In biology, the N number is everything. So
yes,
how did you go about?
So we had collaborators that we worked with where we were able to get brown bear samples. And then we had collaborators in Denmark where we got our polar bear samples through. And that's the way we get it is there's a quota hunt for polar bears in Greenland. And then we were able to, the quota of bears which would have been hunted anyway, we were able to go up and get subsamples of those individuals which are killed. So that's great that we were able to get scientific data out of these individuals as well. So these are data that have been collected over several years in Greenland by some Danish authorities.
What do you think we learn from such comparative approaches?
One nice thing that we found with this project is that we can use data from a non-model organism and a comparative genomic approach to answer questions about evolution, divergence times, demography, selection, and adaptation, which I think was really nice. A lot of the methods that we used have been used for humans and other model organisms, but the fact that we were able to transfer them to wildlife species was very encouraging for future work in molecular ecology, which is the field that I come from originally. I think it was also very interesting to find these genes that have been hugely important for a species that has, within a very short time period, overtaken a new habitat and a new lifestyle, which is very rare. I mean, not a lot of species are able to survive in the high Arctic and not out on the sea ice either. So that was very encouraging.
I'm very curious, what is the next organism that you're going to be looking at?
I think I'm still going to be working with polar bears for a little while yet. But I've worked a lot with African species, so I may double back to them. It's a little bit easier to get samples. But for now, it will be Arctic work for the next couple of years.
Great. Thank you so much.
All right. Okay. Take care.
And congratulations again on your paper.
Yes. Thank you. Thank you.
You can read Dr. Lorenzen's interesting study in the May 8th issue of Cell. I'm Sri Narasimhan, and you've been listening to a Cell Paperclip.